All right, so before we talk about mitosis and meiosis and what they really are, we have to understand what a chromosome is. And a chromosome is just kind of compacted, packaged up DNA that is packaged up with the help of histones and nucleosome proteins. And in humans, we are going to have 23 pairs of chromosomes, and one is going to be the sex chromosome. And this gives us a total of 46 chromosomes. So we get one from our mom and another one from our dad. And humans are known as diploid, meaning that we have two complete sets of chromosomes. And these chromosomes are known as homologous chromosomes, meaning that they have the same type of gene, but just different variants of it. And diploid can be also signified by 2N, okay? And haploid means that we only have a single set of unpaired chromosomes and can be signified by N. And haploid cells in humans are going to be our sex cells. For male, it's going to be the sperm, and then the female is going to be the egg. But in haploid cells, we're only going to have 23 chromosomes, and just N. But for diploid, it's going to be 2 times 23, giving us 46 total chromosomes. All right. So now a good way to look at all the chromosomes, kind of in a human cell, is through what is known as a karyotype, which is shown right here. It's the karyotype. And it's basically just like an image of all the chromosomes that are in one cell. And let's zoom in on this a little bit, and let's focus on just chromosome 1. Okay, so we're going to get one from our mom. Let's say this one is from mom, and another one from our dad. And these two chromosomes are going to be known as homologous. And remember how I said homologous chromosomes have kind of the same type of genes, but different variants, also known as alleles. So take, for example, eye color. Let's say your dad has the gene for brown eyes. That codes for brown eyes. I showed this brown marker here. And your mom has the gene for blue eyes. And those allele combinations will ultimately determine what type of phenotype you're going to have, or what eye color you're going to have, right? So as you can see by this karyotype, we are going to have 22 plus the X and Y sex chromosomes, giving us a total of 23 chromosomes. And how we count these is basically we are going to count them like this. So like this would be 1 right here, and this would be 2, and this would be 3. But I mean, I didn't start from the beginning, but each of those chromosomes separately is going to be called one chromosome. Okay, so now what happens when the cell wants to divide? And this is all about mitosis and meiosis, all about dividing, right? What happens when it wants to divide? So this is another karyotype right here. And this karyotype looks a little different from the one above. And that is because the chromosomes in this karyotype are replicated. All right, and so now if you look at chromosome 1 again, we are going to have a replicated version of each chromosome. And they are going to be known as sister chromatids. Okay, and these are still going to be homologous. These are still going to be known as homologous chromosomes, though. And the way we count these is a little different. So we're not going to count them by each individual sister chromatid. Like this would be 1 chromosome, this would be 2, this would be 3, this would be 4, and so on. And that's not how we count it. We count these chromosomes by their centromeres right here. Okay, it's kind of the thing that's holding those two sister chromatids together. And we say that we still have 23 pairs of these chromosomes and 46 total chromosomes. But in reality, our sister chromatids, we're going to have 92 sister chromatids. And as you'll see when we get into mitosis, with these 92 sister chromatids, when they get pulled apart, each new cell will then have half of that, meaning that it will have 46 chromosomes. Okay, and let's hopefully this diagram down here will help explain that just a little better. So this one to the left here, this chromosome is known as one chromosome, but the one to the right is also known as one chromosome too, because we are counting this by the sister chromatids. I'm sorry, the centromere in the middle, and then those exact copies are going to be known as sister chromatids, right? So counting these chromosomes can be a little confusing, at least it was for me, but hopefully this helps explain it a little better, because mitosis and meiosis is going to kind of be tricky to understand if you don't know how to count the chromosomes, or what homologous pairs mean, or what does diploid mean. So hopefully that helped a little bit. If not, I would recommend going to look at some YouTube videos that help explain chromosomal counting a little bit better. So now let's talk about mitosis and asexual reproduction. Mitosis is the primary way that asexual organisms, along with their somatic body cells, are going to be able to divide. And the cell division process is regulated by what is known as a cell cycle. So here, the first image we have is our cell cycle. And the cell cycle has two main parts, interphase and mitosis. Interphase is where we're going to get that DNA replication, so that we have our we have our 92 sister chromatids that are going to, when they go on to divide, we're still going to end up with the new both cells with 46 chromosomes. And then, so like I mentioned, in mitosis, this is where cell division occurs. And the steps of mitosis, prophase, metaphase, anaphase, and telophase, can be simplified by an acronym known as PMAT. And the steps for that are going to be shown on this diagram all the way to the right here. But your study guide says that you don't need to know that much, all the steps of it. So I'll kind of explain this more simplified diagram that is shown right here. Okay, so for mitosis, we are going to start off with a homologous pair of chromosomes, and we are going to then replicate those chromosomes. Okay, remember that we're going to have actually 46 chromosomes here. We are going to replicate them, and we are going to form, once we replicate them, we are going to have our sister chromatids, the same copy for each chromosome. And we are going to have 92 sister chromatids. So now, after we have all of those chromosomes replicated, we can now kind of finally divide the cell, or do cytokinesis right here. And yeah, there are different phases or different parts and steps in between, and you can look at that further, but this is just a simplified diagram. And so now, once we divide those 92 chromosomes by cytokinesis, basically we're just halving them, just cutting it in half. We're going to end up with two cells that still have 46 chromosomes. Or rather, we're going to have one new daughter cell that has 46 chromosomes, and the original cell that we started with is still going to contain 46 chromosomes. And the whole process of mitosis is basically to create the same type of cell that we started with in terms of chromosomes. So now, if we look at this diagram down below, it just kind of shows meiosis. Briefly shows meiosis, but I'll talk about that more in detail after this. But I just kind of wanted to show the differences between them and the similarities. So like in mitosis, we are going to start off with our homologous chromosomes as well. and then we're going to replicate them, and we're going to get sister chromatids. And after that, we are going to go over to a process known as crossing over, which occurs in prophase 1, but crossing over will basically exchange those alleles with the non-sister chromatids of each homologous chromosome. And this process contributes to genetic variation, along with our homologous segregation as well. And we also have another thing known as I can't remember it. It's called random independent assortment, and all of those will contribute to the genetic variation that we see in our gametes. So after we divide this cell and it goes on to meiosis 2, we're going to eventually end up with our 4 haploid gametes. It can be a sperm or an egg depending on the sex, and these are going to have haploid cells. Remember, have 23 chromosomes, and they are going to be kind of have variation in their chromosomes. So kind of comparing those processes, but I'll talk about meiosis here next in more detail. Now for meiosis and sexual reproduction. Meiosis is a mechanism where we produce gametes, also known as sex cells. In humans, the male gamete is going to be the sperm. Oops, don't know what happened there. Okay. It's going to be the sperm, sorry. And in females, it's going to be the egg. And these gametes, so the one in the male, the sperm is produced in the testes, and the eggs are produced in the ovaries. Okay, so the general goal of meiosis is to kind of go from a diploid cell containing 46 chromosomes and to go from a diploid cell to 4 haploid cells, each containing 23 chromosomes. Okay, so that's kind of just what like a broad overview of what meiosis is. So, and one important thing to know is that we can't forget that in meiosis, we have a great level of variation in the gametes that are produced. And this is kind of what makes each one of us unique and increases our genetic diversity. So, unlike mitosis, where we produce an identical, like a sister cell that's identical to the original, in meiosis, processes such as crossing over, independent random assortment, and segregation of homologous chromosomes all contribute to the increasing genetic diversity in our gametes. Additionally, the steps in meiosis are similar to mitosis, but remember how we had PMAT. Well, meiosis can follow those phases, but it's going to do it twice, and it's going to look a little different. So let's take a closer look at the steps of meiosis and where each of these processes that increase genetic diversity occur, and kind of just track what we get. So let's zoom in on this diagram right here. So this is known as meiosis 1, right? We're going to do meiosis twice. So this part is known as meiosis 1. So here we'll start off with prophase 1. So, like I mentioned before, it's going to have a similar starting process to mitosis, where we have our starting cell that's diploid. But something important here is that in prophase 1, our homologous chromosomes exchange fragments, also known as crossing over. Okay, and they do this to increase the genetic diversity and exchange alleles. So this diagram down here, it's going to kind of show you how that happens. So our homologous chromosomes are going to be right here. And so, right, like as the picture says, they're aligning with each other. But then one of the sister chromatids from the blue chromosome right here is going to cross over with one of the sister chromatids from the red chromosome. Or we can say that non-sister chromatids cross over. Okay, so let's imagine that we had a gene on this blue chromosome here. Let's say uppercase A. Okay, doesn't really matter what that code's for right now, but and on the red chromosome we have an allele that is that is lowercase a. Okay, and basically when these cross over, the alleles are going to swap. So on the blue chromosome, we're going to get the allele from the red chromosome. So right here, the image all the way to the right, I'm going to write that. We're going to have an A on that red part of the blue chromosome, lowercase a, sorry. And then on the other homologous chromosome, we're going to have our uppercase A. Okay, and these are going to be known as recombinant chromatids. And the ones that exchange those alleles, but the other chromatids are going to be non-recombinant. Okay, so that's kind of what happens in prophase 1. So now let's look at metaphase 1, which is the next step. Okay, in metaphase 1, here we see that the homologous chromosomes are now lined up in the middle, similar to that of mitosis, but they are in pairs, unlike a single line. Okay, so let me scroll back up to mitosis real quick. And here, this diagram shows it better. In metaphase for mitosis, these chromosomes are lined up. Okay, but however, they're not in pairs. And in meiosis, those chromosomes will be in pairs. In homologous pairs, as you can see here. Sorry. And the way that these homologous chromosomes line up can be different too, and this is what is known as independent random assortment. Okay, so let's look at a diagram that illustrates independent random assortment a little better. Okay, over here to the right. Independent random assortment is basically where we get a variety of arrangements that the chromosome can be in when they line up in the middle. So let's look at this one at the top here. So during metaphase, we can either have a where both the homologous chromosomes line up like this, where I'm gonna use that, where both of the ones from the dad, okay, let's say blue is from the dad, are going to end up on the same side, and both the ones from the mom are going to end up on the same side. Okay, and this is going to give us our genetic arrangements that eventually will come out like this. Okay, but however, this isn't necessarily what's going to happen all the time. There can be a different arrangement of it. In this lower diagram, you can see that. You can see that the homologous chromosomes for the bottom chromosomes are now switched. So the blue one and the red one are on the opposite sides. Okay, so this red one is on the opposite side of that one, and then this blue one is on the opposite side of the bigger one. And basically what this does is it's going to separate those differently. So one from your dad and one from your mom are going to go to one side, and then another one from your mom and another one from your dad are going to go on the other side. And we're going to get different arrangements, different genetic arrangements of these chromosomes. And random independent random assortment is also going to contribute to our genetic variation that we see in our gametes. All right, so now we move on to the next step, where anaphase 1 occurs. And in anaphase 1, what happens is our homologous chromosomes are pulled to either side of the cell in a process called segregation. And what this ensures is that both of the copies of each gene, one from your mom and one from your dad, are going to be separated equally in the gametes, right? So let's just, like, this one right here can be from your mom, and this one can be from your dad, but they're going to be separated so that each of the gametes will have, they're going to be equally separated into each of the gametes. All right, so now moving on to telophase 1. This is basically where, after cytokinesis, we're going to form our haploid cells. So we're going to form two haploid cells, meaning that they're not going to be in pairs. There's only going to be 23 non-identical chromosomes, right? And basically, that's the first phase of meiosis. And now we're going to use this these two cells that we produced in meiosis 1, and we're going to divide that further in meiosis 2. So let's look at meiosis 2 real quick. Okay. So phases of meiosis 2. Just like meiosis 1, we're going to it's going to still follow that PMAT phases, and but this time we're going to get 4 haploid sex cells. And if you remember, the 23 chromosomes that we get from after telophase 1, right, are going to be right here. These are going to be the ones that come from telophase 1. And they're going to go into prophase 2, where they condense, meaning that we can just see them better. And then they move on to metaphase 2. And in metaphase 2, highlight that here. In metaphase 2, what happens is those 23 chromosomes are going to be lined up in the middle, and they're going to be then separated in anaphase 2. So this is kind of a similar process that we see in mitosis, where those chromosomes are lined up in the middle but not in pairs. Okay, and and they're going to be separated in anaphase 2. Then in telophase 2, we are going to form our 4 haploid cells. Okay, and they're going to be in a variety of combinations. They can be in a variety of combinations. So this is going to be 4 haploid cells. Okay, and something important to note is that we started off metaphase 2 with only 23 chromosomes. So here in metaphase 2, we only have 23 chromosomes in both cells. We don't have like that 46 chromosomes that we would see in mitosis when the cell is going to divide. The cell won't divide in mitosis with only 23 chromosomes because it has to copy them, so that when they divide, each cell is still going to contain 46 chromosomes. However, since we only have 23 chromosomes in metaphase 2 of meiosis, it's not going to divide, right? And this is how we get our haploid cells at the end of meiosis with only 23 chromosomes still. Okay, so one last thing. Let's look at this diagram here of meiosis, and let's try to figure out the possible gamete allele combination, as well as kind of just labeling the parts of where crossing over, segregation, and random independent assortment could occur. So the diagram below shows a female cell with two homologous chromosomes. And we start off with DNA replication for both, okay? And then this diagram is skipping a few steps, and it's only showing this where metaphase happens. Metaphase 1 specifically. But if you remember, what happens before metaphase 1 and prophase 1 is that these homologous chromosomes, or the non-sister chromatids of those chromosomes, can cross over. But this diagram isn't showing that. But just so you know that there's a possibility that these could cross over before that. Okay, and look at the differences in each of these diagrams. So the one on the left is going to show both of these black chromosomes on the same side, whereas on the right, these chromosomes, the black chromosomes are not on the same side, and they're kind of apart from each other, and so are the white ones. So if you remember, what is this called? This is called random independent assortment. Okay, and you can see that here. If you remember what that is, it's basically where those chromosomes can assort independently, and they're going to have different combinations of how they will line up in the middle, and it's not always going to look like, they're not always going to line up together. Okay, so after that, we are going to get past meiosis 1, and we're going to go on to meiosis 2, where basically, well, before we move on to meiosis 2, you should know that these chromosomes that are shown right here and on the same side, or in the same place in the other diagram, are going to get pulled apart, and that is known as segregation. Segregation of homologous alleles. Sorry, not alleles, chromosomes. When they get pulled apart, they're going to look like this, and they're going to have 23 chromosomes in each cell now. And then when they go through meiosis again, they're going to get, the sister chromatids are going to get pulled apart, and then we're going to form our 4 new haploid gametes for both. Okay, and notice how they're going to have different allele combinations based on random independent assortment, because that was the only thing that was changed between these two diagrams. So if you look at this one on the left, we're going to have big A, big B, and little a, little b. But on the right, we're going to have big A, little b, and little a, big B. And that contributes to genetic variation. So if this model were to show crossing over, then it would have even more genetic variation, but it doesn't show that. It only shows that random independent assortment.